Hello and welcome to our TED Talk. Have you ever seen a photo of an actual brain? It looks wrinkly and sort of odd, right? Well, there's a reason for that. We'll get to that in a minute. But first, let's discuss a very famous brain. Albert Einstein is arguably one of the most intelligent humans to have ever lived. You'd think a really smart person like that would have an extraordinarily large brain. Well, turns out size isn't everything.
The autopsy of Albert Einstein's brain revealed that not only was his brain not any bigger than the average human brain, it was actually smaller. As showcased by the story, we often associate having a large brain with intelligence. Perhaps intelligence is not so much determined by the size of the brain, but rather by its ability to rapidly and efficiently transfer and integrate information from different regions of the cortex. But how is this achieved in practice?
Looking at these images of a mouse and a human brain, one thing in particular stands out. The human brain is very wrinkly compared to the smooth surface of the mouse brain. The wrinkled part we look at is called the cortex. This part of the brain controls a multitude of wide-ranging cognitive, motor, and behavior tasks. This wrinkling of the brain is referred to as cortical folding and is believed to be an important contributor to our ability to process information. For one, folding of the cortex means that there is a large cortical surface area where much of the information processes takes place. Besides, because of cortical folding, neurons in the cortex are closer together, which allows for faster and more energy-efficient transfer of information.
But what do we actually mean with cortical folding? Cortical folding is a relative ingenious solution to increase the surface area of brain matter that can be packed into relative constant skull volume per species. It enables us to house more neurons while keeping the volume minimal.
There appears to be an evolutionary benefit to cortical folding, and it is clear that not all animals have highly folded brains like us humans. So how exactly does cortical folding occur? As usual in science, multiple ideas have been proposed to explain this phenomenon mechanically, each trying to cover a different aspect of cortical folding. However, it seems that in recent years, most scientists have agreed that cortical folding is at its base the result of different growth rates of the inner and outermost parts of the cortex. As the brain develops, the different layers of the cortex expand, however, they do so at different rates. In general, the outermost part of the cortex, called the cortical plate, expands at a significantly faster rate than the underlying layers, causing the cortex to fold in on itself.
If this were all there is to it, and cortical folding was entirely regulated by faster tangential expansion of the outermost layer of the cortex, we would expect to see somewhat random patterns of cortical folding at regularly spaced intervals. We would expect the brain to look somewhat like this. And while you're looking at this image, you might think two things. One, that looks like a brain. And two, who would want to eat that? And while this cake might look like a human brain at first glance, if we compare it to an actual brain, you'll see that there are significant differences. Most notably, there is no repeating pattern of folding. Instead, there seems to be a more complex hierarchical pattern of deep fissures, or primary fissures, subdivided by smaller secondary and tertiary fissures. Now, as it turns out, this hierarchical pattern of cortical folding is already present during development, where primary folding occurs at an early stage, followed by secondary and tertiary folding. Now, in addition to this, the primary folding pattern is largely conserved among individuals of the same species, and is even quite similar when comparing phylogenetically related species, such as the Siberian tiger and the clouded leopard. Together, this indicates that the mechanisms underlying cortical folding must be tightly regulated by differential gene expression. And indeed, studies have found thousands of genes which are involved in the development of the brain that are differentially expressed across different areas of the cortex. Interestingly, many of these genes showed an alternating pattern of high and low expression, which very closely resembled the folding of the cortex.
The reason the cells in your brain are able to contort and bend in such elaborate ways is because they possess a very special physical property called viscoelasticity. You can think of this property by thinking of a ball of Play-Doh. It seems to hold its shape just fine, but if you set it on a flat surface where it is exposed to the effect of gravity for a long time, the ball of Play-Doh will look like it's sinking into the ground. Brain cells are also like this. They can hold their shape up to a certain point, but after that point, they will begin to change shape, take on a new form that will help them compensate for new mechanical constraints under which they have been placed.
Is this then the cradle of intelligence? Is this cortical folding a better marker for intelligence than brain size? Let's have a look at other species' brains to get more of an insight into this. Before we do that, it's good to consider that intelligence is actually a very human-centered concept. What we find intelligent is what we find very similar to us. However, should we use the human brain as the reference when we consider intelligence-related concepts such as cognitive abilities, if even one of the most intelligent people to have ever lived did not have a particularly special brain?
While cortical folding is a general feature of mammalian brains, different degrees of folding equate to different levels of intelligence.
Dogs, for example, have folded brains. While they can be trained to perform complex motor tasks on command, higher order cognitive processes are a bit too far-fetched for them.
So far we've been discussing human brains. Humans achieve their intelligence by folding their brains more. Other species have different strategies. Consider, for example, the crow. Research has shown that crows are able to recognize and remember human faces. They've also been documented to use tools for complex problem solving. The intelligence of crows has been recognized by humans for centuries, so much so that many ancient cultures have revered them as gods or messengers of the gods, like in Norse mythology. In summary, unlike humans, the brain of a crow is very smooth. They are fairly intelligent animals, and they're able to do this by a strategy called neuronal density. Simply put, they are able to pack more neurons into their very small brain surface area, even more than most primates.
A number of corvids have been found to use tools in the wild. A study showed this in an experimental setup in which they demonstrated how rooks, a member of the corvid family, use stones to raise the water level to reach a floating worm. They used 4 captive rooks and exposed them to 3 different experimental setups, varying the height of the water, giving them the choice of using large or small stones, and including a control tube in which there was sawdust instead of water. All 4 subjects solved the problem with just the right amount of stones that were necessary. They also rapidly learned to use large stones over small stones, and that the sawdust cannot be manipulated. This behavior demonstrated a flexible ability to use tools. Note that this is not learned behavior that was taught to the birds by the experimenter. These are just the natural instincts of the rooks.
Different evolutionary streams have resulted in various brain shapes and structures that all attempt to achieve more or less the same goal: to produce an animal that is capable of adapting to its own environment and solving complex problems without having to drastically alter the total volume of its brain. There is much more we still don't understand about the intelligence of other animals. To do this, we must further explore the process of cortical folding.